before I forget. So this is our first seminar or webinar of our series of plasticity seminars. So they're based on the French network UGDR Plasticité Phénotypique. Right, and so I just want to give a reminder for those of you, you cannot hear us. Someone cannot hear us.
Christian.
So I want to just give a brief technical introduction for those of you who are not used to that. So this is the webinar mode of Zoom. So you can, during the talk, you can ask your questions in the Q&R or Q&A tab in the bottom if you want. And then people can vote for the questions they would like to be asked. And then at the end of the talk, you have an opportunity to directly ask your question to Antonia and interact. If you haven't had time to write in the Q&R, then you can just raise your hand or something like this. What else did I want to say? Yeah, those of you who were at the last UDR meeting, you know that I've become a survey fan. So now there's a survey tab here. I'm curious about how you heard about this talk, because it's been advertised by different ways, by different means, Twitter, emailing list and stuff. So I'm curious to see which one worked for you. So if you can just click on what's your answer, that would be great. And now, so yeah, just the point is, if you have questions, please type them in your Q&R and not in the chat or discussion thing. That would be there. So now I just leave Jean-Michel introduce our speaker for today.
Okay, hello. So it's a great pleasure to welcome Antonia Monteiro for a webinar for GDR on phenotypic plasticity. Antonia studied in Portugal. She did her PhD in Edinburgh with Paul Breckfield on the evolutionary genetics and developmental basis of eyespots morphology in butterfly wings. She then did a postdoc in the USA at Harvard, and the second one at Leiden in the Netherlands. She was then assistant curator of entomology at Yale. Then she became assistant professor at Yale University and later at the National University of Singapore. And since 2020, she has been professor at the National University of Singapore and the Yale National University of Singapore College. And during all these years, Antonia has used butterflies as evo-devo models to study the development and evolution of pigmentation patterns and the mechanisms underlying phenotypic plasticity and its evolution. She has used particular pigmentation patterns present on wings, eyespots, as model of development and phenotypic plasticity. And Antonia has also investigated the adaptive role of wing eyespots through behavioral experiments. And with a group, she has developed and used genomic tools, drugs, transgenesis, and CRISPR mutagenesis to perform functional experiments in butterflies. She has published remarkable papers on the mechanisms underlying phenotypic plasticity of eyespots and its evolution, and also other aspects of wing patterning, such as wing vein or the production of structural colors by wing scales. So thank you very much, Antonia, for giving this talk. We are all looking forward to listening to you.
Thank you very much, Jean-Michel, and also Luis Miguel and Vincent for this invitation. Let me see if I can start by sharing my screen like we tried before. Okay, so it's a great pleasure for me to be invited for this webinar, and I'll give you a little bit of the recent. I'll tell you a little bit about some older work and more recent work that we've been doing on the origin of phenotypic plasticity of eyespot size in nymphalid butterflies, in particular in satyrid butterflies. So we are all interested in biodiversity and how organisms acquire their beautiful colors and shapes and patterns and so forth. And we've all learned that natural selection is usually the cause of creating organismal adaptations to their environment. So those organisms that are better adapted to their environment tend to survive and produce more offspring. However, organisms don't live in stable environments. Many organisms live in environments that look quite different, for instance, during the winter and the summer months. And they have evolved not just one adaptation, but they evolved multiple adaptations in order to better survive in each of these different respective environments. So here, for instance, the Arctic hare has evolved a white pelt that produces beautiful background matching when the environment is white, full of snow. And so is this ptarmigan and so forth. So these organisms are able to change their pelt and their feathers throughout their lives and then change them back again in order to continue to be better adapted to their background environment and so forth. Now, insects don't usually go back and forth, but they also adapt to their respective environments. But once they do so, they usually extract cues from the environment where they are developing as larvae. And then they use those cues as predictors of what the future selective environment of the adults is going to be like. And they produce a particular type of adaptation. In the case of butterflies, it's often in the case of different color patterns. So for instance, the spring form or the summer form of an African butterfly, they can look quite distinct and different, but this is the exact same genome that is producing these two very different outputs depending on seasonal variation. Here we have beetle horns that can produce long horns or small horns. This is in fact not a seasonal adaptation, but this is plasticity in response to a maternal provision of nutrients in the form of the size of a dung bowl where these eggs were laid. We also have not really seasonal polyphenisms here, but in the case of these locusts, you have the gregarious form and the solitary form that form in response to mechanical cues. So aggregation cues lead to these different forms of the locust. And also an example of Daphnia, also not a seasonal cue, but in this case, presence or absence of chiromones. So these are molecules produced by fish predators in the environment that lead the Daphnia to produce a bigger helmet to better protect them against fish predation. So these types of phenotypic plasticity in invertebrate are usually irreversible. And they are produced in response to environmental cues. So these cues, as I said, inform the genome that a change in the environment is about to take place or is actually taking place. And then somehow the genome evolves an architecture that allows these environmental cues to alter development and the final phenotype of the organism. And so what I study are these phenotypic plasticity in a butterfly that lives in an African seasonal environment, in particular a dry season and a wet season environment, so in the African tropics. And that these two environments actually provide quite different selection pressures for this particular species that I study. So it's called Bicyclus aninana, and it has these two seasonal forms. So the wet season form has these marginal eyespots that are quite large and striking, and it lives during the wet season in Africa. And the dry season form produces these reduced eyespots, also a reduced white band in the center of the wings, and it lives in the dry season in Africa. So this is the same genome that gives rise to these two different forms. And we can mimic the production of these forms in the lab simply by changing the rearing temperature of the larvae. So if we rear larvae at high temperatures, we produce the wet season form. And if we rear the larvae at low temperatures, we produce the dry season form. And temperature seems to be an important cue that is telling development of the larvae that it should either switch to produce the wet season form or the dry season form. So I've have three topics that I've decided to approach in this webinar. The first topic is just to give you an idea about the ecological function of eyespot size plasticity in the species, and then a little bit about the physiology and the genetics of how temperature, in fact, is regulating eyespot size. And then finally, the last topic is to try to understand how did this happen? So what are the steps, the genetic steps that took place for temperature to actually have this effect on eyespot size regulation? So how did this plasticity originate on a phylogeny of nymphalid butterflies? So the first topic is just to show you that this plasticity seems to be adaptive. Early studies that were using vertebrate predators, such as these flycatchers, done by Anna Luttinen et al. In the lab of Mapes, she sort of put the dry season form and the wet season form of the butterfly in cages in the lab with interaction with these predators. And what she found was that if the cages had been sort of matted with dry leaf litter, so to simulate a dry leaf environment. This is what's represented here by the brown background color here. These flycatchers had a tough time detecting the dry season butterflies, which were well camouflaged against the background. But they could easily detect the wet season butterflies, and they would predate on them first. So this is what the rank order of predation here means. So they would be eaten first, and only later would they detect the more cryptic dry season form. So these experiments show that the dry season form seemed to be adapted in terms of evading predator detection against the dry leaf environments. But when you sort of had the cage filled with green leaves everywhere, there was really no difference in the order of predation of these two different types of butterfly. And it was not clear at all what the large eye spots were doing in terms of providing any sort of adaptive advantage in terms of escaping predation. So the role of large eye spots was not entirely clear. Katie Pruddick then came into my lab. She was a behavioral ecologist interested in, expert on sort of dealing with mantids, praying mantises. And she decided to test how invertebrate predators actually look at eye spots and how they attack the two different seasonal forms of this butterfly. So I'll show you a little video of her work. And here what she's doing is looking at interactions of praying mantises with dry and wet seasonal forms. Sorry, let me see, I have to press this arrow here. So the two forms are here, and we bred them in the lab, and we also bred from egg cases the Chinese praying mantis. So here's a mantis that is attacking a dry season form. And you see that it scooped up the head and the thorax of this butterfly. It did not care at all about the wings. Here's another attack in slow motion. Dry season form. No eye spots or small eye spots. And that's the point of attack. So the body, the head, the thorax. But now you can look at, so the mantis takes away the wings and then chews on the body. Now here's how the attack works when the eye spots are present. So this butterfly escaped the attack with torn wings, and it's actually this butterfly on the left that was the attacked butterfly. So the rips all happen in the area of the eye spots. And here's another one in slow motion. So the attack is directed at the wing margins rather than at the thorax and the head. So it looks like when we have invertebrate predators, they do get fooled by the presence of these eye spots on the wing margins. And here's the data in sort of quantitative terms. So here's a wet season form butterfly, and most of the attacks, 68% of the attacks were towards the hind wings where the eye spots were present. Whereas in a dry season form, most of the attacks were towards the head, the thorax, and the abdomen. So having eye spots seems to be a good adaptation to have when you're being attacked by an invertebrate predator. And what might happen in the field is that vertebrate predators are present all year round. And by the way, here's the sort of the data that shows that when the mantid attacks a wet season form, they actually escape much more often than when the mantid attacks a dry season form. So having the mantid rip the wings is okay because the butterfly can still fly away and lay eggs and so forth. So what we think is happening here is that perhaps different predator guilds in different seasons may be selecting for this alternation of eye spot sizes, meaning in terms of vertebrate predators, it's always better perhaps to have no marking on your wing that attracts attention, because they will just gobble you up regardless of where the eye spots are there or not. But in the wet season, there are perhaps a higher number of invertebrate predators. And for their sake, it might be better to evolve these deflecting marks, these large deflecting marks on the wing margin in order to get ripped preferentially on the wings rather than on the body, and to allow the butterfly to survive a little longer and to lay eggs and so forth. So this is just the introductory bit about why we think this eye spot size plasticity is an adaptation in these satyrid butterflies. So now looking a bit more mechanistically about how does rearing temperature during the larval stage actually regulate the size of an eye spot? So we rear them in the lab, as I said. At 27 degrees, we produce the wet season form. At dry season, rearing them at 17 degrees, we produce the dry season form. So the two questions we asked was, when during development is eye spot size plasticity being determined in response to temperature? And what is happening during those temperature sensitive periods at the physiological level? So to answer the first question, what we did was a temperature shift experiment where we reared the animals from larva to pre-pupa to pupal stage at predominantly at one temperature. So in this case, the green bars mean that most of the developmental period of these butterflies were at 27 degrees. But for 2 days, for 48 hours, we would have a small interval of time where we would shift the temperature of the animals to a different temperature, so to the dry season temperature, and then shift the animals back into the main temperature, like 27 degrees. And we did this, and we used this 24-hour shift. We placed it at different time points during development. So we used it during the larval stages, at different time points during the prepupal stages, as well as the pupal stages, to try and pin down what was the most temperature sensitive stage in terms of a 2-day window that was responding, that had an effect on eye spot size. And we did the exact same experiment where predominantly the temperature throughout was low temperature, except for a small window again of 2 days when the animals were placed at 27. So, and what we found was that in both of these types of shift, so predominantly at low temperature or predominantly at high temperature, with just a 2-day window at high temperature, the wandering stage is the most temperature sensitive stage. So here, it's when a 2-day window at the opposite temperature almost completely reverses the size of the eye spots. So makes them quite large. If during the wandering stage, the animals sense high temperatures for 2 days, that's sufficient almost to turn them into a wet season form. And the opposite is true also. If we rear them throughout at high temperatures, but during the wandering stage, we place them at low temperatures, that produces a dramatic shift in the size of the eye spots, in this case, downwards. So we wondered if this hormone, this molting hormone that had been implicated before in the control of eye spot size regulation, was shifting, was whether there were different levels of this hormone at that specific stage in development. So this hormone had been looked at before we did this experiment, just in the pupal stage, but not in this particular wandering stage when we discovered was the actual most temperature sensitive stage. So we decided to look at the wandering stage, whether there were differences in titers of this hormone, the molting hormone, and whether there were differences also in the receptor of the hormone, the ecdyson receptor, and whether these differences, if present, whether they were functional. So we first measured levels of the hormone throughout the different periods of the wandering stage. So these are wandering stage larvae at 20% wandering development, 40, 60, 80, 100% in prepupal stage. And in fact, we found that the wet season larvae had higher titers of this hormone relative to the dry season larvae. There were also differences between males and females. So females in both seasonal forms had still higher titers than males, but on average, the green lines were above the brown lines at the end of the wandering stage of development. And then we examined also at that stage what was happening to the receptor for this hormone, so the ecdyson receptor. And we looked at wings that are still inside the wandering stage larvae. So these are small, about 1 millimeter in diameter wings that you can dissect out of the body of these larvae in the second thoracic segment and the third thoracic segment. 100% wandering stage, there's an exponential growth. So the wings are growing quite a lot at this stage, and we dissected these late wandering stage larvae, and we stained them for using an antibody against this protein, this receptor, ecdyson receptor. And what we found was that there was expression of the receptor, particularly at high levels in the cells at the center of the future eye spots in these hind wings that are particularly plastic and sensitive to temperature. So not only do we have high levels of the hormone in the wet season form at this moment, we also have presence of the hormone receptor in the cells at the center of the eye spot and at the same stage in development. We did dissect both dry season and wet season wings at this stage, but we could not find any difference in the expression levels of the receptor. So the only asymmetry in the developmental system that we could detect was an asymmetry in the levels of the hormone, but not in the levels of the receptor of the hormone in both dry season and wet season wings. And so to test whether this asymmetry was functional, what we decided to do was to manipulate ecdyson receptor signaling by manipulating both the hormone levels. So in this case, using dry season wandering stage animals, we injected them either with vehicle or we injected them with higher levels of the hormone with 20E, and we were able to obtain a shift in the size of the eye spots. And we did the opposite experiment, which was in animals reared at high temperature, so producing the large eye spots, we injected them with vehicle, or we injected them with a hormone receptor antagonist molecule, which is a competitor. It's a molecule that actually binds the ECR receptor and prevents the 20E that is present in this form from actually binding. And so it's a competitive molecule, and it prevents ECR signaling from happening. And this led to a reduction of eye spot size in these forms. So these functional injections of hormone and hormone receptor antagonist molecules show that this plasticity is being mediated. So eye spot size plasticity is being mediated by different levels of the hormone 20E that is being produced in response to different temperatures, produced at different levels at different temperatures, and is regulating eye spot size at that stage in development. So the answer to question 2 of this talk is that temperature regulates eye spot size via changes to titers of 20E and via ECR mediated signaling at the specific wandering stage of larval development. And finally, we wanted to understand how this complex mode of regulation of eye spots originated on a phylogeny of butterflies. So there's actually 2 views on plasticity in the field, as far as I understand it. So some people, and I belong to that group of people, I guess, Bradshaw, Saravia, De Jong, Steve Stearns, Nancy Moran, they've all proposed that plasticity is most likely a derived adaptation. It's a product of natural selection itself, and it helps organisms survive in these variable environments, and it adapts organisms to each of these different environments that they live in. So this is their view. However, there's a sort of an alternative view out there, proposed primarily by Massimo Pigliucci, by West Eberhard and others. And their view is that perhaps plasticity is ancestrally present in most organisms, that most organisms by default, they are flexible, they are sensitive to the environment, they have non-canalized developmental processes, and most organisms will actually produce some sort of a response of their phenotype in response to variable environments. And this type of flexibility that they ancestrally have allows them sometimes to adapt to novel environments and colonize novel environments and so forth. So I can sort of place those 2 views on plasticity in this type of format, in this scheme, which is the ancestral view claims that plasticity is old, is present in most traits, is ancestral, and it helps organisms colonize and adapt to novel environments. And then once they are in a fixed environment, perhaps they just lose it and they become canalized to either express one form or the other form of the plasticity. Whereas the alternative view is that it is definitely a derived adaptation. It needs specific mutations to actually be to occur in organisms, and it's definitely not something that is ancestrally present in most organisms. So we wanted to address this question for our system of eye spot size plasticity. We wanted to know if organisms that perhaps have never lived in a seasonal environment, so butterflies that have evolved, for instance, in the tropics, that have in more the equatorial tropics, so different from Malawi, where Bicyclus aninana originates from, which has a more seasonal type of temperature environment. We wanted to know if butterflies that had never been exposed to seasonality in their environment actually were ancestrally plastic or not. And so we performed a large comparative study. And we used, and this was done primarily by Shivam. He was a graduate student in my lab in Singapore. And Shivam primarily went to a butterfly farm in Malaysia that reared a lot of tropical equatorial type species. And he reared them in the farm at 2 different temperatures, at usually a 30-degree temperature and a 20-degree temperature. And he reared about 13 different species, many of them tropical, equatorial, tropical. Some of them outgroups, like they don't even have eye spots. They have these spots on the margin of the wing, or this nymphallid that has spots but not eye spots. And this isn't a complete outgroup outside the nymphallids. Papilio, so it has these red spots at the edges of the wing. So we tried a few outgroups with just sort of wing spots. But then we tried a series of nymphallid butterflies with proper eye spots as well, from these different regions of the world. So the little maps on the side show where these butterflies live. And so a lot of them are equatorial, but some of them have a sort of a broader distribution. Bicyclosanninana is over here, so it's spread out throughout Africa. Morpho is a South American butterfly. It's quite spread out north and south out of the equator. So they all have eye spots. So the graphs I'm going to show you are of the following type. So I'm going to show you graphs for each one of these species that Shivam reared at 2 different temperatures in this farm in Malaysia. So what he's plotting here is the log wing area and log CU1 area. Now CU1 is a sort of the jargony name for this specific eye spot. So it's the one that occurs in the cubital 1 sector of a butterfly hindwing. So we're always just measuring this one spot or eye spot size in all of these species. And the blue dots represent the animals that were reared at cool temperature, so usually in this case 20 degrees, and the red dots represent the animals reared at 30 degrees. So the reason why we plotted log wing area against log eye spot area is that often there is a positive correlation between the size of a butterfly and the size of its wing markings. So we wanted to then regress wing size out of this equation. And we wanted to just, for instance, take what is the size of an eye spot for a given wing size. So if you just cross these regression lines at a particular wing area, if you take a vertical sort of cross section, the graph on the right is going to give you what the average eye spot size is for the same wing area across all of these butterfly species that I'm going to show you. So wing area is taken away, and all what we're looking at is to see if for a specific wing area, the animals reared at high temperature have a larger or smaller spot than the animals reared at low temperature. And in this case, there is a very small slope. So the reaction norm in terms of eye spot size in response, or spot size in response to increasing temperature, is almost flat, or in this case, the spots are slightly smaller with increasing temperature. And this is what the data looks like for all these other species. And what we find, and if we just look on the graphs on the right, is that most species are not plastic, and they do not actually change the size of their wing markings, spots or eye spots, in response to temperature. When they do, they actually do it in an opposite way to the way that Bicyclus, which is this species that I've been talking about for a while now. So Bicyclus increases the size of their eye spots with increasing temperature. All these other butterflies have either a flat reaction norm or have an opposite type of reaction norm, which is they decrease the size of their eye spots with increasing temperature. And so when they decrease the size of their eye spots, it's still sometimes a small decrease compared to, for instance, the large increase that we see in Bicyclus aninana. So this is rather interesting and surprising. And it shows that on a phylogeny, the particular type of plasticity that we encounter in Bicyclus aninana is unique in this group of species that were reared. And it's a derived adaptation. It's a derived trait in this case. Most of the other species in white, they are not plastic, or they have a slight decrease in slope, and that's what this red arrow means. And this decreased reaction arm seems to have evolved perhaps twice, here at the base of this group, and here again in these outgroups, Danainae, which are one of the subfamilies of nymphalid butterflies. So Shivan, in addition to looking at what was happening in terms of the response of eye spot size to temperature, he looked at what was happening to the titers of the 20E hormone in response to temperature. So he dissected, he took hemolymph from all of these animals at the exact same wandering stage that we had examined before in Bicyclus aninana butterflies. So he extracted hemolymph from the blood of these animals at two different temperatures. So again, at the low temperature and at a high temperature. And he found that the titers of the hormone usually raised with increasing temperature. So for most of these species, they were plastic in this specific physiological response. So the hormone levels increased with increasing rearing temperature in the blood of these animals. So they all seem to have this, what I would call a pre-adaptation to rearing temperature. The titers of an important hormone that regulates eye spot size in this species also increases in all species in response to rearing temperature. Now, the other component that Shivam examined that we needed to be present is we needed to, for the eye spots to be sensitive to this hormone. So we needed to know whether all of these eye spots, whether they had that type of sensitivity, meaning whether the epidermis, so the cells at the center of these pattern elements, express the ecdyson receptor. And what we found was that the outgroup, so any of these species here that don't have eye spots, so they have simple spots at the base of the nymphalids, or they have these simple spots at the margin that are not really true homologs to spots or to eye spots, or these papilio spots. None of these species have ecdyson receptor expression in their markings. But all of these species that have eye spots, with the exception of this Junonia almana, which does not express ecdyson receptor at this stage in development, they actually all had the ecdyson receptor expression in the center of these eye spot pattern elements. Now, even though they all had ecdyson receptor expression, it looked like they were not using it to regulate the size of the eye spots, because as we saw previously, only Bicyclus aninana actually was responding to ecdyson signaling, ecdyson receptor signaling. But to test this further, what Shivam did was he took an outgroup, which we knew should not respond to ecdyson receptor signaling because it did not have the ecdyson receptor expressed in its spots. And we took 3 other species that had ecdyson receptor, but we knew that these species were actually having a negative type of reaction norm plasticity. So we wanted to know if for some crazy wiring, the ecdyson receptor here, instead of increasing the size of eyespots. When we manipulated it, actually decreased the size of the eyespots. But what we found was that this, these species, if we inject 20E, or if we inject the antagonists, cucurbitacin B, they simply do not respond to ecdysone signaling manipulations, whereas bicyclus, again, is the only species that responds to ecdysone signaling manipulations. So what happens is that even though these species have the ecdysone receptor and could potentially respond to ecdysone receptor signaling, they don't. So whatever it is that is important in creating a connection between ecdysone receptor signaling and eyespot size control happens in this species, but is not happening in these species. So what does this all look like? It looks like, so this is all what we found mapped on a phylogeny of all these species. And what we have is a sort of the evolution of a complex adaptation in Bicyclus aninana and potentially in all these other Bicyclus and satyrid species, which we did not examine in the study in detail, but we knew already from other work, from other labs, in particular Paul Breakfield's lab, that all these satyrids, Bicyclus, Michaelisus, Heteropsis, they all have the specific type of plasticity that Bicyclus aninana has, which means the eyespot size increases with increasing temperature. All these other species have this other form of plasticity or no plasticity at all in response to rearing temperature. And this other form of plasticity seems to have evolved twice, perhaps here once and perhaps here once independently. And the form of plasticity that we were after, this Bicyclus aninana form of plasticity, required these three components of plasticity to be there. So it required plasticity of 20 titers, which seems to be a pre-adaptation and seems to be present in all these species of butterfly. It required the expression of the receptor in the cells at the center of the eye spot, which was an earlier step, an earlier mutation that happened in all butterflies that have eye spots. And then it required, in addition, for sensitivity for the eye spot gene network to gain sensitivity to active ECR signaling. And that seems to be only a property of satyrid butterflies and not of any of the other butterflies that do have ECR receptor, but they don't have sensitivity of the eye spot network to that active receptor. So in our case, how does plasticity originate? So this should have been topic 3. So it looks like it's not an ancestral trait. Plasticity in eye spot size, in particular, when eye spot size increases with increasing developmental temperature, is definitely not present in ancestral species of nymphalid butterflies. It's a very derived adaptation that took multiple steps and millions of years to evolve and to be produced. So it's definitely something that is not present in a majority of butterfly species with eye spots. What's actually quite interesting from the studies that even though we knew we were working with butterflies like this Junonia almana butterfly, it's an nymphalid butterfly, lives in Southeast Asia, lives in Singapore. We know that this butterfly, for instance, can produce dramatic seasonal forms in nature. So we can photograph it like this in the dry season, and we can in Thailand, for instance, and it looks completely different in the wet season. This butterfly is not doing this type of plasticity by regulating, it's not using temperature as a regulator of its plasticity. So temperature produces a complete flat reaction norm when we rear these animals in the lab, which means that in these other places in the world, like Thailand and so forth, that have equally dramatic seasonal plasticity in eyespot size, they are using completely different environmental cues that need to be examined. Perhaps they're using photoperiod, humidity, we don't know. But all these instances of plasticity that are as dramatic as the ones that we see in the African satyrids, they are regulating plasticity in completely different ways that need to be examined in future. So in conclusion, in Bicyclosanninana, eyespot size plasticity seems to be an adaptive trait against both vertebrate and invertebrate predators. Temperature leads to changes in 20 titers at the wandering stages of larval development. And these changes, these different titers, they are essentially regulating eyespot size in these satyrid butterflies. And satyrids, but not other nymphalid butterflies, they are using ECR-mediated signaling to regulate eyespot size. And this seems to be a derived stepwise adaptation acquired by this group of satyrid butterflies over millions of years of evolution. And the people that did this work, most of the work in part 3 was done by Shivam, a fantastic graduate student in the lab. The behavioral work with mantids was done by Katie. I don't know why she disappeared. Ashley also helped obtain titer data for the hormones, and Xiaoling was involved in doing some of the earlier stains for examining the expression of the receptor for the hormone in the wings of these butterflies. These are the funding agencies, and I'll take any questions. And thank you for your attention. And I'll stop sharing for now.
Okay, thanks for the great talk. Very, very clear and lots of food for thought. So there's already lots of questions. I want to ask Mathieu Joron, your question.
Hi,
you were the first to ask a question, then it means it's solved. Does it mean that you got the answer yourself or something went wrong?
Oh no,
you did a mistake, Vincent.
Yeah, exactly.
Okay,
yeah,
so Mathieu, I think you can ask your question. Let's see if I can turn on your mic. In the meantime, people can sort of start to vote for the questions if there are questions that they like or they would like to see asked. Otherwise, we'll just go through them in the order where they appear. Mathieu?
Can you hear me?
Yes. You can turn on perhaps your webcam if you have one. I don't know if it works. Perhaps you cannot.
I'm not sure.
Okay, yeah.
I can't turn on.
Okay.
Webcam. It seems to be locked. Great talk, Antonia. Thank you very much. And I didn't know the word by Shivam. It's amazing. My question is about the first part on the ecology. I was wondering why, or whether you knew if the wet season forms tend to be more active. So could this be part of the reason why the mantids might miss them more often, because they would react more quickly to attacks and only get their wings ripped, basically.
Thanks for that question. Actually, that's an excellent question. And Katie, who did this work, was wondering about that too. And what she did was she did a follow-up, which I did not talk about, but she actually cut out the wings of wet season forms, and she glued those bits of eyespots, just the strip at the edge of the wing. She glued them in the bodies and on top of the wings of dry season animals. And she did the same experiments with the mantids. And what she found was that it was not behavioral differences between the two forms that led to the different fitness differences. The fitness differences could be explained pretty much solely by the appearance, by the eyespot presence on the wings of these butterflies. So that was a sort of an interesting follow-up that addressed that issue of the behavioral differences.
Okay, great, thanks. I should have known that maybe.
It's okay. It's all in that paper in 2015.
All right, so I'm not even sure, in fact, in this version of Zoom that we can vote for the question. So we just go to the next one on the list. So it will be alphabetical order who sets the order. So Arnaud, if you can, sorry, Arnaud, where is he? Just turn on your mic.
Can you hear me?
Yes.
Okay, so thanks for the talk. I was wondering if there was any kind of polymorphism in plasticity, like all individuals respond the same way to the temperature, or are there different kinds of genotype? I mean, is there some kind of quantitative variation in plasticity?
So that type of work was explored a little bit by students in Paul Breakfield's lab, maybe 10 years ago, maybe 15 years ago. What they did, I remember the work of Viltus. He was trying to select for flat reaction norms in the lab, and he kind of obtained that a little bit. So he was able to select for bicyclosaninana wet season forms that stay kind of wet season throughout all temperatures, and lines that were selected at different temperatures, but trying to produce dry season form throughout all temperatures. And those experiments were somewhat successful, showing that there is some variation for the reaction norm to evolve in this species of butterfly. Does that address your question?
Yeah, sure. Yeah.
All right, then there's a question by Ariel. SB.
Yes,
yeah, now you can turn on your mic, I think.
Yes, can you hear me now?
Yes.
Okay, good. Yes, thank you for an interesting talk. So I was wondering about inferring the ancestral state of the plasticity, and in particular, when you measure the reaction norms, the range of the environmental variable that you're looking at. And in particular in Aniana, for example, it seems like you're using either 17 or 27 degrees, whereas for some of the other species, you use 20 and 30 degrees.
Yeah.
So do you think that you would see a different plasticity patterns if you had changed the range of the temperatures? And related to that also, what is the penetrance of the temperature effect on Aniana morphs? So if you go to 20 degrees, do you then see higher frequencies of the other morph, or?
So what happens with Aninana is that it's pretty much a continuous response of eyespot size to temperature. So if you increase the temperature gradually, and this has been done a lot in the lab of Paul Breakfield. So if you rear at 17, 19, 23, 25, 27, you get a kind of a sigmoidal response. You don't get a continuous response to temperature, but you get kind of a sigmoidal. The sigmoidal inflection point happens at around 23 degrees. So you have kind of dry season-y forms, but they are not all the same dry season. The eyespot size increases a little bit, and then it shifts a bit more strongly at around the 23 degree inflection point, but then it keeps increasing kind of gradually beyond that point. The other species, I think, might have similar responses, but we did not have the time to examine them in detail. And the reason why we shifted them to 20 degrees and 30 degrees is because we tried rearing them first at the exact two temperatures we used for Aninana, but 17 degrees is pretty lethal to some of these equatorial species. So many of them would not make it at 17 degrees. We had a lot of mortality. So we needed to shift the low temperature to a little higher temperature. So we shifted it to 20. And in order to keep the distance the same, so to have a 10-degree separation for all of our species, we decided to shift the high temperature as well to 30. So that was the reason why we have slightly different temperatures. But most of those species are reared at 20 and 30, and bicyclists, if reared at 20 and 30, would show the same type of plasticity as it would show now as it shows at 17 and 27.
Yes, thank you. Can I ask a follow-up to that?
Yes.
So what is how do you know how the temperature modulates the hormone?
Yeah, so those are sort of questions we don't know yet the answer. So we don't understand how exactly outside temperature impacts maybe the glands in the head that produce ecdyson in the larval stages. We don't know how that works. We also don't understand the connection of what's downstream of ECR signaling. So what is this active hormone bound to its receptor? What is this compound, which is an active transcription factor? What is it binding to in the eyespot gene network? So what are the downstream targets? So this is something that a current PhD student in my lab is examining, potentially with ChIP-seq.
Thanks very much.
We know something in the middle, and we don't know how it all starts, and we don't know what this hormone compound does. So those are sort of still open questions.
I have a question.
Yeah.
It's known that there is a signal coming from the center of the presumptive eyespot, which is important for patterning the eyespot. And is it known whether this signal or signals, if there are several of them, are downstream of the ecdyson receptor?
Okay, that's a very good question. And we want to know the answer to that question badly. What I can tell you is that we have very exciting data where we've done CRISPR against the ECR receptor, and we have lost eyespots completely, not just made them smaller or larger. We've lost them altogether. And so this is very interesting because it might be that the origin of eyespots is connected to the recruitment of this plasticity gene to the eyespot center cells, because this plasticity gene is also known as a pioneer transcription factor that can open chromatin in specific ways and can create a completely novel type of epigenetic landscape in important genes. And so if this recruitment of the ECR receptor is an essential gene for eyespot development, it might have been essential as well for opening up chromatin and allowing the recruitment of this much larger gene regulatory network to the eyespot centers as well to have taken place for the eyespots to originate for the first time in the wings of butterflies. But this is all pretty preliminary data, but we would like to know the answer to that as well. Yeah.
Yeah, that's very interesting.
Yeah.
So there was another question by Vincencio. I think you can turn on your, Right.
Hi, Antonia. Thanks very much for the great talk. Actually, you already answered the question I had, but I had a different one. That's all right. So how do you think ecdyson receptor expression evolves between the species or has evolved between the species that do show plasticity in its expression versus those that don't?
Yeah, I mean, that's something we would really like to know. So we don't know if the expression itself, I mean, there's two things. So we have ecdyson expression in a bunch of species with eyespots. All of the species with eyespots have ecdyson expression, but not all of them, just the satyrids, as I've shown you, seem to respond in the specific way that Bicyclus does. So ecdyson expression evolved at some point. Perhaps it's connected to the origin of eyespots, perhaps. But plasticity is something else. And so the evolution of plasticity has something to do either with evolution of specific targets for ECR. So imagine evolution of binding sites for the ECR receptor in distillus or any of these other eyespot genes, Spalt, etc. So those genes, perhaps in species that don't have plasticity, don't have ECR binding sites in their regulatory sequences. And in satyrids, the species that have evolved plasticity, perhaps these genes have evolved binding sites for an active ECR receptor complex. So that's one way. Another way is that they may have particular microRNAs that facilitate some something in the network to get turned on or turned off. We really don't know. I mean, there's many possibilities that are just completely open.
Great, thank you.
Yeah.
Okay, I had a question on the evolution of plasticity. So I found it really interesting to investigate whether it's derived or ancestral. But I was wondering whether you had looked at that at a smaller phylogenetic scale within the Biciclus genus, for example, and contrasting species with different ecologies or.
So we haven't done that, but people like, I guess, Vicentio and Oscar and other people that have worked on the evolution of plasticity in bicyclus species, they have looked at that. From what I gather, all of the bicyclists that have been reared in the lab so far at 2 different temperatures, they all have the exact same reaction norm as Bicyclus aninana. So all satyrids, as far as I know, have the exact same response. So we think that the evolution of this specific reaction norm and sensitivity to temperature in this specific way is probably a feature of satyrid lineages, of the ancestor perhaps of all satyrids or most satyrids. We haven't pinned it down to that level yet. But anything else like the morphos, which are the close relatives, as you know, to the satyrid lineage, they don't have that reaction norm anymore. So the morpho butterflies are the sister lineage to satyrids, and they don't have it anymore. So somewhere along the satyrid lineage is where I would place the origin of this specific type of plasticity. But I don't know where, yeah.
There was one question by one guy, Tan, but I think he left. So I think I'll ask my own question then. I think this probably won't be very precise, but I was very, I found it very striking how you can decompose these different levels, these different bricks that are involved to have this plastic response. So this also seems like something a bit complicated that could be disrupted at any of these levels, in fact. And there can be contingency at each of these levels. Like, for instance, just for some reason, this satyrid group had this thing that made them able to not only have the receptor in the right place, but also be able to connect it in a way or use this information. And this really kind of opens up a program of research. I don't know how many people can do it with these details, but I think finding the complexity of hierarchy levels involved in different types of plasticity perhaps is one way to understand how readily it can evolve or be lost, etc. So have you been thinking a bit more about this, considering your case, and if it can be generalized to other things, or I don't know?
Yeah, I mean, I do like the idea of decomposing. I mean, I like the idea that we knew our system in one species sufficiently well that we could decompose it at least in these 3 components, which we did that Shivam did in this comparative work across butterflies. So I think any system, if you already know a few of the components that work together to produce the plastic response, I think it's possible to decompose any system and look at those individual components separately, because this involves a developmental gene, like a receptor, being present in the right cells to make them sensitive to a hormone. So if this type of knowledge is present for any other plastic system, like insulin receptors, for instance, in horn beetles, or any other type of hormonal mediated system that involves a particular receptor, particular hormone, juvenile hormone receptors, I think it's possible to decompose a system in these different parts and then look at it in a comparative sense. How we're doing it now is as I mentioned, we're going to look at ECR targets, and we're going to do it also in a comparative sense. So we're going to look at ECR targets in Bicyclosan inana and in a species that does not have plasticity, but has the ECR expression, to see if there are different targets that have evolved that have allowed this connection between this hormone receptor complex that is lurking in these species that have it, but it has not yet connected in a functional way to regulate size of an eyespot. So why is it not connected functionally? What is it that needs to evolve next? So that's what we're going to be doing in a comparative sense as well.
Yeah, I think it's very, I mean, it's very convincing in your case that you have this hormone-mediated process, and it's simple that you can decompose this. But to me, it also asks lots of questions in terms of evolution, which genetically, which things can evolve more readily, and like the hormone seems to be something that's organism level, so it's just sensitive to the environment and transmit a signal to everything, and then the receptor brings the organ receptivity or things, something like this, specificity, sorry, if you see what I mean.
That's true. I mean, you can always decompose. People have always thought that when you have a hormone-mediated system, it's pleiotropic and you cannot break it apart. But of course you can, because a hormone-mediated system, only the cells that respond to the hormone will become sensitive to it. So if you don't want sensitivity to a fluctuating hormone, you switch off the receptor of that hormone in those cells. But that's not the only way you can do it. You can switch off the binding sites in the genes, in the regulatory regions of genes that bind the hormone receptor. You can switch those off, and then you lose sensitivity as well. Or you can create more sensitivity to a fluctuating hormone by evolving more binding sites to that hormone complex in the cis-regulatory elements of genes.
Yeah, so you can couple also the level of response.
Yes, you can do it gradually, you can do it abruptly. For instance, in our own system, we know that the anterior eye spots on the wing, they're not as plastic as the hind wing eye spots, where the predators attack primarily. So those forewing eye spots, which are used for sexual signaling, even the ventral ones, they have done that. They have removed the receptor from their centers. So the front wing eye spots have just switched off the receptor, and they're not plastic as much as the hind wing eye spots are. So that's how they did it, even within one organism, within one different wings.
Different instances of the same trait. Yeah. So there was another question by Mathieu. So I don't know how much time you have, you know, in this group. Usually we like leaving time for discussion. So if you have time to take another question by Mathieu?
Sure, I have all the time.
Okay. Mathieu, tu peux allumer le micro. Hein?
Yes. Thanks. Perhaps you've answered that question already, but I was wondering, I mean, all the species seem to be plastic in the same kind of way to temperature with respect to the level of ecdyson. So I wondered why you think ecdyson is responding this way. Is it related to something very common to growth rate in all the butterflies, or regulating molting, or?
Yeah, that's a good question, Mathieu. I've never really started delving much into that trying to figure that out. But it could be what you said. It could be something to do with growth rate, that it's a molting hormone. It may just be very sensitive to environmental temperature. It regulates development slower if it's lower temperature, faster if it's higher temperature. But I really don't know. I cannot, I think this deserves a lot more study, and we haven't done it. So I don't know if it's even more broadly, like in crustaceans or all arthropods, do they have that type of plastic response? Yeah, I don't know. It needs to be looked at.
Okay, thank you. Thanks very much.
So some people are raising their hand. I don't know if they want to ask something more.
Yeah, if I can.
I mean, I think you can, yeah.
Okay, yeah, thanks.
We'll
so,
stop at 3:30 or something if it's too
okay,
long, but
good.
we're like having discussions, so that's the point.
Yeah, that's nice. Yeah, I thought it was very nice that you've identified the developmental stage at which they are actually sensitive, which seems to be now a particular time period within the wandering stage. Is it something particular about the wandering stage at why they're sensitive? Do you know?
Well, I guess, let me think. Why is it particular about the wandering stage? It's the stage when definitely the titers are peaking. So the other stages in development, the hormone levels are pretty low in the blood. So the hormone levels really peak and then they come down, crashing down again right after pupation. So that makes them, the fact that these animals are growing at different temperatures doesn't seem to impact. So going back to Mathieu's question, actually, I'm kind of answering it myself now, but it looks like temperature, outside temperature doesn't really do much to the hormones at all until these sort of before molt stages. So before each molt, there is a peak of hormone, of 20E hormone. And again, just before pupation, there's this big peak in the wandering stage. And why it happens, why is eyespot size sensitive at that stage? Maybe just because by chance eyespots are also being determined, the eyespot centers at least, are being determined developmentally at exactly that same stage. So the two things kind of go together. So it was a cue, was an asymmetric cue that was already there in tissues or in the blood of animals at the time when eyespot centers were being differentiated. So I think evolution just took what it had. It took those asymmetries that were there, in this case, physiological asymmetries, and used them to regulate eyespot center differentiation, because it actually does regulate the size of the eyespot at that time. The expression of the genes in the center become reduced to a very small group of cells in the dry season forms at that time, or you get a pretty big focus, a pretty big group of cells that becomes the eyespot center if you experience high hormone levels. Maybe it was just serendipity, the fact that you had an asymmetry at the time when the trait was being determined.
Do you think it's because also you see the involvement of ecdyes in wing polyphenism in PA aphids, for example? So you see the sort of repeated use of the same hormones being used.
That's true. Although in Pierrids, I'm not sure if Andrew Stower and other people that have worked on this plastic, if they've done the temperature shift experiments and shown it to be larval rather than pupal stage, the sensitive period for the spot size plasticity in Pierrids. I think they have not yet done the temperature shift, so we don't really know precisely when in development Pierrids respond to temperature. But yes, you're right. They also, I think, seem to respond to 20E. Actually, I'm not sure who did the experiments. Has anybody done experiments with 20 injections, or I'm not actually sure they have. I cannot remember.
Yeah. Thanks a lot.
Yeah.
I have a question for the cells at the center of the presumptive eye spot, which are emitting a signal. Is it known if the amount of signal is different in the between the different seasonal forms?
So we haven't looked at that, although we definitely should. But the first data that I remember about this was the data from Paul Breakfield's lab. It was a Nature paper from 1996, where they looked at distillus at the time when distillus was the only gene that had been associated with eyespot development. And what they found was that the expression domain of distillus in the early pupal stage was already different between the two seasonal forms. So the dry season forms had a small cluster of cells expressing distillus, and the wet season forms had the bigger cluster of cells. But of course, we know that distillus is not a signal, it's not a morphogen. We now have antibodies against armadillo, which is also not a signal, it's a signal transducer of the wingless signaling pathway. And we could look at that, and we haven't, between dry and wet season forms.
Because I was thinking about all the grafting experiments where it was shown that if you take the presumptive center of the eye spot, you can induce an ectopic eye spot by grafting. So you could also take the center of the wet season or the dry season and put them in the other form to see whether you induce a small or a big eye spot.
That's a great experiment, actually. I'll ask a student to do it.
That would be...
I like it a lot. We've never actually tried doing grafting experiments between seasonal forms, but that would be actually quite informative, because it would tell us that at the time we do the grafting, which has to be like between 3 and 5 hours after pupation, which is when the epidermis is still attached to cuticle, and so you can cut a nice square of cuticle and attached epidermis. Yeah, it would be informative, because it would tell us that by that time in development, the cells at the center have been determined to be either dry or wet season, irrespective then of the response environments that they are grafted into. Yeah, that would be very nice, actually. Thank you. I think I'll try to get the student to do that. Yeah, these are very tricky. It takes forever for the students to learn how to do these cuttings, but I think it's worthwhile.
All right, so I think maybe we can stop here, because we already had quite a nice discussion. So thanks again, Antonia. This was really a great talk, going from the selective pressure to the mechanism and evolution. So really, really nice start for our, this webinar series. And actually, so the next one, it's not sure if it will be the next one, but there will be one on March 16th by David, who is in the audience, I see today. So this is promising to be a really great talk too, so don't miss it. What we'll do is that we'll advertise it on our mailing list, which is our network, but we'll also keep all these people who registered today we'll send the emails to them too, and we'll advertise it also on Twitter, etc. So, so this video, the video for your talk will be available on YouTube pretty soon, so you can share it for people who couldn't attend today. Okay. Thanks again.
Thank you very much, Jean-Michel and Luis Miguel and Vincent, for the invitation. It was a pleasure.
Thank you very
so
much.
much.
Bye-bye.
Really nice.
Bye-bye.
Bye-bye.
Bye-bye.